Greetings, students. So today we are going to start covering a topic that historically is a pretty tricky one, but I'm confident that with some work before class and then some good work in class that you will all be able to overcome and kick butt on these chi-squared tests. Okay, so chi-squared tests are an example of statistics, and to just remind us why statistics is important, I have a fun little analogy for us. Why do we need all this darn math in our science? Okay, well, let's imagine a similar analogy. Let's say you lend one of your friends $20, and then the next week you all go out to lunch and your friend is like, hey, since I owe you $20, I'll buy your lunch. Okay, great. So let's say the bill comes and your lunch comes out to $19.95. Well, you lent your friend $20. Would you say that's close enough? Okay, what if your bill came out to $18? What if your bill came out to $15? So I hope you guys can all get where I'm going with this. At some point it feels like, no, that's too different than $20. Like I lent you $20, not $15. And so that's sort of what statistics help us do. They help us figure out when is something close enough. And so a lot of this is going to come back, hang on, there we go. So in biology, how we're going to use statistics, biologists use statistics to analyze their data. And what that means is they'll be able to say, take two groups where they have done some manipulation and measured some outcome, and then statistics is going to help scientists figure out, are those groups different enough to be remarkable? And if they are, we are going to use this very fancy and jargony scientific term, which is statistically significantly different. And that just means that we use statistics to help us say, this difference seems to actually be important. Okay, so how do we do that? Well, the way that we get to this statistically significant difference is we have to test the null hypothesis. So this is something we learned about way back in the first week of school, so let's try to remember this again. But the null hypothesis is just that there is no difference between two groups. The way I like to remember it is null means zero, so it's the hypothesis that there is zero difference. So let's go through and do some examples, just sort of looking at some of these different types of null hypotheses. So in this little definition, they're saying the null hypothesis assumes that there is no relationship. So in other words, no difference between two variables. So let's think about, in this example, the null hypothesis they've come up with are cats show no preference for food based on shape. So in this case, the groups we would be comparing would be fish shape food and triangle shape. So we'd have some fish-shaped food, some triangle-shaped food. We'd give cats bowls of both, and then what we would measure would be food consumption, food eating. That's not great English. And we would get some sort of data, and then we'd be able to use that data to test the null hypothesis. And here, the null hypothesis would be, cats eat the exact same amount of triangle food as fish-shaped food. Okay, let's do another one. So in this example, the null hypothesis is that plant growth is not affected by light color. So in this case, the groups we would be comparing would be plants grown in yellow light versus plants grown in pink light. And then we would grow our plants in these two different colored light, and we would measure plant height or plant growth or something like that. And then we would take those data and we would use them to run a statistical test to test our null hypothesis that plant growth is not affected. In other words, that there is no difference between yellow light and pink light. Okay, let's do one more. So in this null hypothesis, we're saying that age has no effect on musical ability. So what would be the groups we would be comparing, and what would we be measuring in this experiment? So the groups would be young musicians versus old musicians, and we would be measuring musical ability. And we would use that data, compare the two groups to test the null hypothesis that young musicians have the same ability as old musicians. Okay, so that's a quick and dirty review of null hypothesis. So how do we actually test that null hypothesis? When we have that data, how do we actually figure out, are the two groups different or are they the same? If the null hypothesis is true, that means the groups are going to be the same. Because remember, the null hypothesis is that there is no difference. So if our null hypothesis seems to be correct, then that means there is no difference between the two groups. If our null hypothesis is false, that means the groups are different. So to look at that in sort of like a graphic form, if we had data that looked like this, in this case, group A and group B have the exact same value, the groups are the same, we would say we fail to reject the null hypothesis. And we say fail to reject because we don't like the word except. It's like too strong in statistics. So sometimes I'll say except in quotes just to help us remember that we're not rejecting. So except the null hypothesis. So in other words, those two groups are the same. On the other hand, if we had data that looked like this, these data, the groups are quite obviously different. So in this case, we would reject the null hypothesis. These groups are not the same. Okay. So how do we actually decide, are we going to reject or fail to reject the null hypothesis? Well, to that we need p-values. Remember, we talked about this at the start of the year too. So p-values are what we're going to use to actually put a numeric number on reject or fail to reject. And the way I like to remember how we use p-values is with the rule of thumb. So if p is greater than 0.05, so thumbs up, greater than, that means we are going to accept, or in other words, fail to reject the null hypothesis. So in this case, that means the groups would be the same. If our p-value is less than 0.05, thumbs down, less than, that means we are going to reject the null hypothesis and our groups are going to be different. Okay, let's practice. Let's imagine that we do some sort of statistical test and we get a p-value that is 0.53. Would we reject or fail to reject the null hypothesis? Yeah, okay, so in this case, 0.53 is greater than, so greater than 0.05. Remember, greater, thumbs up, that means we are going to accept or fail to reject the null hypothesis. In other words, the groups are the same. Okay, let's try another one. So in a different experiment, we have a p-value that is 0.043. In this case, should we reject or fail to reject the null hypothesis? Yeah, in this case, we are going to reject the null hypothesis. So, same thing, 0.043 is less than 0.05, less than, thumbs down, that means we reject the null hypothesis, and we would say the groups are statistically significantly different. Yikes. There we go. Okay, so this is all stuff that we covered earlier in the year, so hopefully some of this is coming back to you a little bit because we are definitely going to need to remember all of this in this next unit. Okay. So let's talk about what the null hypothesis is going to be in this unit in genetics. So thankfully, in this unit, the null hypothesis is always going to be the same thing. In genetics, we will always use the same null hypothesis, and it comes back to this idea of Punnett squares. So when we do a Punnett square, it's a prediction. So we're saying, okay, if up here, these are the possible gametes. Oh, I can't find the mouse now. So if we're saying, up at the top, those are the possible gametes that would be created from the father, and then on the side, the possible gametes from the mother, then we know because of probability that we should get roughly these outcomes. That's what a Punnett square tells us. So in this unit, our null hypothesis is going to come back to this idea of Punnett squares. So essentially, the Punnett square is going to be what we expect to see. So in this example experiment, let's say we cross two plants, they're both heterozygous for yellow color, and we end up getting 100 offspring. Well, we could set up a Punnett square, so AA times AA, and we would set up that Punnett square and we'd see, well, we expect 3/4 yellow and 1/4 white. And since we had 100 offspring, that would mean 75% of 100. We would expect 75 yellow flowers and 25 white flowers. But then we do the experiment and what we actually see would be 70 yellow and 30 white. So the groups we are comparing here in the genetics unit are always going to be the same two groups. We're always going to be comparing the expected outcome, so like how many yellow versus white did we expect to see versus the observed. And so our null hypothesis is going to be that there's no difference between those two. So in genetics, the null hypothesis is always going to be that there is no difference in the phenotype ratio of the expected offspring and the observed offspring. In other words, in genetics, our null hypothesis is going to be that what we got from our Punnett square is going to be the same as what we actually got. Okay, so in order to actually answer this question, to test this null hypothesis that expected is the same as observed, well, we need to get a p-value. How do we calculate a p-value? So we need to use a statistical test, and this is the only statistical test that you will need to calculate for AP Bio. It's something called a chi-squared test, and we are always going to use this to compare observed versus expected. That is the point of chi-squared tests here in AP Bio. So we are using this chi-squared test to test our null hypothesis. Notice that I'm saying chi-squared, not chi-squared. I know that's how it looks, but it's chi, like rhymes with bi. That has to do with that little Greek letter. It looks sort of like a fancy X. So this is the formula for the chi-squared test. This would be on your formula sheet, both for the final and for the AP exam. You also will have all of the different values spelled out for you, but let's go through it together. So first we have the chi-squared value. This is a really important thing to note because a lot of students will make a mistake here, which is so painful to see. This test is the chi-squared test. We are calculating the chi-squared value. What we want from this is this entire part. So what I want you all to remember, if you're taking notes, I want you to write this down and box it really big, is don't take the square root. Don't take the square root. This is a very common mistake and one that is so painful to see because you'll do like everything correctly and then take the square root at the end and it's like, no, you're so close. So it's the chi-squared test because we are solving for chi-squared. Just chi is nothing. Great. Okay, so that's the first part. That's what we're solving for, this chi-squared. Let's look at the other parts of this formula. So this little Greek E looking symbol, that means sum. That means we're going to add multiple things together. We'll talk more about that in a second. And then, O, that's the observed value. So that would be like how many yellow flowers did we actually see? And then E, that's going to be the expected value. Those are going to be values that we need our Punnett square. This would be like from the Punnett square. Okay, and that's the whole formula. It's not so bad. So let's go ahead and work through an example problem. Okay, so let's use our same example where we have these expected results that we have calculated using our Punnett square. So here was our Punnett square. Remember, we had 100 total offspring. So we can use, well, we know we should get 75% yellow offspring, and if we had 100, that means we would expect 75 yellow. But we actually saw were 70 yellow. So we're going to use those numbers to calculate just this part of the formula. So let's do that. Okay, so for yellow, the observed we saw was 70. minus the expected was 75 squared, and then divided by the expected, which is 75. And if you do that math and plug it into your calculator, you will see that you end up getting 0.333. I like to round at the thousandths place for this. It just makes sure that you're not going to cut off some important decimals, but you don't need to go out to like 10 decimal places or anything like that. Definitely don't round until the very last step in these problems. Okay, so that's just this value for yellow, and now let's do the same thing for white. So for white, we had 25 expected and 30 observed. So in our formula, O minus E, so 30 minus 25 squared divided by expected, 25. We plug that in and we get 1. Okay, and then remember we're going to take the sum. So the sum just means we're going to add. So we're going to add those two together, and we will get that our chi-squared value, so that's chi-squared. Our chi-squared value is 1.33. Okay, so this is one way that you could do this problem. If you are a very confident math student and you have worked with sums before, then this way will probably work well for you. But what I would recommend pretty much everyone do actually is a different method, which we're going to call the table method. It's doing the same thing, but we're just going to do it in a more step-by-step way, so you won't accidentally make like a math mistake, and it just makes life a little bit easier. So this is the table method. It's still the exact same formula, so we're still using this up here, but now we're just going to kind of plug the numbers into this table step by step so that we won't get confused. You will oftentimes see this table printed for you on exams, like I'll print it for you on our upcoming formative. There even was an AP exam question on a short response where they made you fill out this table. So it is important to kind of understand what the steps of it are, even if you feel like really good about the formula already. Okay, so let's go through. So first we're gonna just write down the observed numbers. So yellow, we observed 70 yellow, 30 white, and then down here I'm gonna do the sum. So what was our total offspring? 100. And the reason that's important is because that's what we're gonna use to calculate the expected values with our Punnett square. We've already done that up here. So our expected yellow was 75 and white was 25. Okay, and then in the next column we're just going to do observed, so O minus E. So this will be 70 minus 75, which will be negative 5. And then in the column after that we're going to do O minus E squared, so this will be negative 5 squared, so this is 25. And then in the last column we'll do O minus E squared divided by E, so just the previous column, 25, divided by E, which was 75, so we get 0.333. Okay, now we'll do the same for the white. So O minus E is 30 minus 25, so that's going to equal 5, and then we square it, 25, and then O minus E squared divided by E, 25 over 25, so that gives us 1. And then finally down here we're going to take the sum, so we're going to add them together, 1.333, and that equals our chi-squared value. There we go. As you can see, it's the exact same as before. It just kind of helps you go through the process step by step so you don't miss anything. And you really want to make sure you show your work on these problems, because on both the final and on like AP questions, you would get points for doing all of these things in a systematic way, even if you made like a math mistake at the end. So make sure you show your work. Okay, so now we've got that, my chi-square value. Okay, so now we have that chi-square value, and remember the whole point was we were trying to figure out what's the p-value, so we need to remember what's that p-value. Well, to find the p-value, we're going to need to use this chi-squared table. The chi-squared table has a lot of stuff in it, so in here, this box, these are all the possible chi-squared values, and they are corresponding to these p-values. And then we have this one extra little layer of stuff we need to understand, and that's this idea of degrees of freedom. This is just telling us what column in this table do we need to look at. So we need to calculate the degrees of freedom, which are going to be the number of groups you were just comparing minus 1. So in our experiment, we had yellow and white were like the two phenotypes we were looking at. So we had two groups, minus 1. So our degrees of freedom would be 2 minus 1, which is 1. So you would just be looking at this column in the chi-squared table. Okay, so great. If you remember, our chi-squared value was 1.333. We're going to look in this column and do something. What are we going to do? Okay, so remember, in biology, we're using that p.05 as our significance cutoff. And so we're going to look in this chi-squared table to help us figure out if our p-value is greater than or less than 0.05. So our chi-squared value in this was 1.33. And we're going to look just in this 1 degree of freedom column, and we're going to see where would our chi-squared value land within that column. If it's going to land somewhere where our p-value would be greater than 0.05, turns out that's above the table. Greater, thumbs up, we accept, fail to reject the null hypothesis, and our groups are the same. Groups are same. So in other words, observed equals expected. If the p-value is less than 0.05, thumbs down, we reject our null hypothesis. Observed does not equal expected. They are different. Okay, let's go through that one more time. Okay, so in our experiment, our chi-squared value was 1.33. So we are going to look, remember we had our degree of freedom. We just had 1 degree of freedom because we had 2 groups, 2 minus 1 is 1. We're going to look here in this column and ask where would 0.133 fall here? Well, as we're going, okay. So 1.33 would be like somewhere up here, right? So that means 1.33 would associate with a p-value. Our p-value is going to be greater than 0.05. So greater, thumbs up, we accept the null hypothesis. Our groups would be the same. In other words, observed equals expected. Let's write that out in paragraph form. Okay, so this is sort of a paragraph stem that I will give you guys, and I recommend you get used to seeing your results of chi-squared test written this way. In fact, if you were taking notes right down, I would write down this whole paragraph and just get used to seeing it and get used to restating it. So the conclusion that we would say at the end of this entire chi-squared journey would be the calculated chi-squared value is 1.333. We got that from the table. With 1 degree of freedom, this gives us a p-value greater than 0.05. Therefore, we fail to reject, or we accept, the null hypothesis, and that means there is no statistically significant difference between the observed and expected phenotype ratios. So in other words, what we thought we would get from the Punnett square is pretty much what we actually saw, and we were able to make that conclusion because we got that chi-squared value of 1.33. Okay, let's do one more. So since you've written down that paragraph, I want you to, in the little question that's about to pop up, I want you to type out that same paragraph and tell me what conclusion would you draw from this experiment. So let's assume we have a totally different experiment, and in this experiment there are 2 degrees of freedom. So since there are 2 degrees of freedom, you're going to be wanting to look in this column. And our chi-squared value is 10.53. So what conclusion would you draw from this? Okay. And that's it. That's chi-squared tests for now. We are going to, like I said, do so much more practice with this in class, so it's okay if you're feeling like, yikes. But if you aren't feeling super confident, I would highly recommend you watch this very good video on chi-squared from Bozeman Science. Just the more exposure you can get to this before you come to class, the easier this concept will be, and it's already a really hard concept, so getting a little bit more exposure will only help you out in the future. So I highly recommend that you take some time to watch that video if you can. And that's it. So please just make sure that you look over your notes before you come to class, And if you are still feeling confused after class, then please come to tutoring this week, because this absolutely is going to be on our quiz this week, and it will totally be on the summative and the final, and it's something that you know is coming, which means that we know we need to figure out how to do it, and then we can crush it and get those points. It'll be easy points if we all can figure it out. Okay, so that's chi-squared tests, and I hope it wasn't too painful, and that's it for now. See you guys later.